Thank you for being here at the first afternoon session. So the title of this session is pretty long. It's Barley End Uses from Food and Feed to Malting, Brewing and Novel Products. And that's partly due to the fact that we had to combine two sessions here. There was originally one session on malt and another session on food and feed. So barley as a food is one of my own interests, and that's why I think this is a really important session as well. I've recently been involved in projects where we had clinical trials, nutritional clinical trials with barley among diabetes patients. And we actually found that barley is really good for people with diabetes. It really controls the blood sugar way better than you can do with wheat. So we definitely there see some potential for the use of barley as a healthy food. So I'd like to introduce the first speaker, and that is Professor Henrik Pedersen from Aarhus University.
Thank you, Kim, and thanks to the organizer for inviting me to give this talk here at this conference. So we learned this morning that around 80% of the barley that's grown is actually used for feed. So I think I will use around 80% of my time on talking on barley feed. Then I have a few examples at the end that on products we could also talk a little bit about. I will I decided to take two examples. And it's examples where we have actually taken it from all the way through the chain up to the feeding experiments to see and get the final evaluation of our ideas. I will start with an example. The first example is something we've been working with for many years. I actually started with this enzyme in my PhD, and then at that stage we didn't know anything about it, and then we have sort of developed it, and now we are able to sort of take it into some lines which can be used for feeding purposes. And then the next example is something that we have not we didn't work that much with it before we started, and got an idea that this could perhaps have an impact on the feed quality of the grain in poultry and also in pigs. I have to confess that I will use a couple of examples from wheat, and that is because the feeding trials is mostly done in poultry, and they're not super happy with the barley. So that's so there we have used wheat, but barley is very much in focus of what we're doing. So the first example is on. It's actually about this compound called phytic acid, which is sort of the main storage compound of phosphorus in the cereal grain. Up to around 80% of all the phosphorus stored is stored in phytic acid. It's very distinct localization pattern in barley. It has around 90% in the lower layers and 10% in the embryo. It chelates minerals in the digestive tract, so it's actually the most important intermedicinal factor of micronutrients in feed, but also in food actually. It needs to be degraded by an enzyme called phytase in order to be available for uptake in the intestine. And this is sort of the first example of a phytase that degraded phytic acid. It goes from IP6, which is phytic acid, and then one phosphate is released, and that is actually the critical steps to go from P6 to P5, and then it's further degrades down in this case to P3. So 3 phosphate groups are released. So and that is what required in a feed situation, because it needs to be graded. Otherwise, the phosphate cannot be uptaken by the animals. If we look on the cereal grain, then we deal with basically 2 types of phytase activity in the grain. We have sprout phytase activity, so that is induced during germination. It's not so relevant when we talk feed, because we don't normally germinate our grains before using them as feed. We could, but we don't. And then we talk about the so-called mature grain phytase activity. So that is a phytase activity that is synthesized during grain filling and then stored in the mature seed. And that is directly the one that is used when you is active when it's used for feed. Among the cereals, there are huge differences in the level of mature grain phytase activity. The non-Triticea species like rice and maize and so on basically has no mature grain activity, whereas the Triticea tribe cereals, where barley belongs, has some activity in the mature grain, but usually it's not enough. Interestingly, rye has a high activity, I'll get back to that, and also some of the more exotic cereals like monococcum and Triticum Egilops, has a high activity, but I'll get back to that. But as it is, unless you do something then with the phytase activity, then in a diet, then just in this case, this is from a pig diet, and you actually supplement with feed phosphorus also, so because it's simply not impossible for the animal to digest the phytic acid. So we have a very low deposition of phosphate in the animal. In this case, it's around 30%, so 70% of all the phosphorus is passing directly through the animal, and that is not really a good idea. Phosphate is a non-renewable resource and so on, and then there are environmental issues and so on. So basically, and that's been known for many years, we need more phytase activity in order to digest phytic acid. And that was something we worked with for years to figure out what is actually the phytase that is needed. And it actually took us quite some years, and this slide is sort of a summary of, I don't know how many years of research, but so we now know that the phytase in the plant is a purple acid phosphatase phytase, and that is completely different from what we thought to start with. We know how it's located. We can even do it. We express it in the pig. We can even, it's a metalloenzyme. It is actually purple, and we can express it and do everything with it. We also know the active sites and so on. So this has taken us long to figure out what to target, which we wanted to improve the phytase levels in the cereals. Recently, we also got the crystal structure of it, which just gives us some opportunities in case we wanted to do some base editings, which I think we will. But there are two PAP phytases in barley, also in wheat. We have the A type situated on chromosome 5, and then we have the B type chromosome on chromosome 3. We know that the A is the one that is synthesized during grain development, so this is the one that gives rise to the mature grain phytase activity, whereas the B is the one that is de novo synthesized during germination. If we look on the promoters of these two, then they are completely different. So the PAP A, so that's the one synthesized during grain development, consists of a set of motifs that are sort of, well, it's typical for genes expressed during grain filling. In particular, we are interested in this region here with a so-called GCN4. It's a motif that you also can find in promoters of the storage proteins and the SQN. So there's a conserved region here, which I'll get back to, whereas the promoters of the B holds motifs from genes that are induced by an example, gibberellic acid, and so on, responsive elements. So certainly a gene that is expressed de novo during germination. So, but we had to sort of figure out about this. So the PAP A is our candidate, and here we have made a knockout with a CRISPR. If we knock out the PAP5A, so we had to sort of study the potential of this particular phytase, so we knocked it out. If you sort of make a knockout, it simply doesn't have any phytase activity at all. So it's gone, and this is different mutation, but if you make sort of a substantial deletion in that region, then you basically have no activity left. On the other hand, we could also, in this case, use it for accelerating the activity. And this example, we used it as a cis gene, and we even done field experiments and stability studies and so on, and we have stacked the number of cis genes in a Barley. In this case, we go from sort of 0 additional copies up to 6 additional copies, and we done that by crossing of different cis gene events spread over the different chromosomes actually, and then we made double haploids to double and so on. And there is a correlation, at least in this range here, in the number of the phytase gene inserts in the genome and then the phytase activity. So by stacking them, we could increase the activity, in this case, to up to 7,000 units per kilo. So that's sort of quite interesting. But this was cisgenesis. So we were screening a lot of different accessions and cultivars and species and so on, and we knew that the rye had a very high phytase activity, and that's partly because it has an extra phytase gene in the genome. It's sort of a natural cis gene compared to the wild type barley, but it also has, in a specific region, we found out that in this region I was pointing at earlier with the GCN4 SKN, so there's an odd base palindrome, so repeat there, we saw some specific mutations there. And interestingly, the ones that we could see in the monocopium and the rye, which has both high activity, has a very specific mutation in a specific place. So we have been screening like crazy, all sorts of wheat and barley and so on, and done a lot. One day, a technician came in and say, "I have tested this wheat here we have from a population somewhere from, and it's very high. It has a very high activity." And this is one up here. And I said, it can't be true, you have to do it again. So she came back a couple of days later, and this activity was still high, very high actually. So that was very interesting. So we took that and identified that this particular mutant, which we call the hi-fi mutant, actually had a mutation in exactly the same place in the promoter as we see in the rye and also in the monococcus. So that we decided to take all the way to feeding experiments. So we did a multiplication with help from Francisco Barro in Spain, in Andalusia, where they can grow 3 generations per year of wheat, and we got, well, from one plant to our field here, you see enough material so actually doing a feeding study. And with help from Nottingham Trent University, who are very good at this, we did with 180 broilers feeding experiment, or they did, we didn't, where we were placing. So we're having here, so minimum P is sort of adding inorganic P, and then supplement here, the treatment is with a commercial phytase which you can buy. But then we replaced the standard wheat with 33% high-fi, 66% high-fi, and 100% high-fi. and saw a increase in ELP and calcium digestive sort of growing with an increased amount of high-fi wheat. This is the total P and calcium digested in the feed, and it's really competitive, and actually also if you replace 100%, it's actually better than using the commercial phytase. So this was for wheat. We are now pursuing sort of to get the same mutation in barley, and with help from the excellent platform Trait Trommings used, developed by at Carlsberg. I think we will hear more about it, I guess. We have sort of targeted the same region and achieved a serious mutation, not exactly currently the same as we have in the wheat and in the rye and in the monococcum, but we have a mutation close to it, and it's just to demonstrate here that it's a very delicate balance. So if you take so we have 3 mutations called LIM 5, 7, and 11, and 13, sorry. And you see here, this is a progression of phytase activity. So days after germination. But if we just take here at day 0, this was done in planet. Then we have an activity here in planet in wild type, and then we have a specific mutation in LIM 5. So you can see it has an increased activity. It's not exactly the same level that we expected, but it is certainly, that region is confirming that this region is very active, important for this. But you also see in this one here, it's super delicate. If you just go upstream a few bases and make a mutation here, it's a single mutation, then you have no activity at all. So it's a super delicate balance to find that, but it confirms that this particular region is super important for expression. We are in the lab, and this is not the phytase gene, but this is just to pay your attention to poster number 13, where Zelalem is doing promoter bashing on one of our genes. And then here he has, so go to that poster, which we want to hear more, where he's targeting the Cossack region by this way, mutating the areas, but also in his poster there's also a base editing result. But basically he can increase the expression of target gene by mutating these genes, but that was sort of extra bonus for you. So example number 2 is something that we didn't actually expect should give something when it started. It's also about feed enzymes. So feed enzymes are supplemented to most poultry and also pig feed in Denmark and also in other countries. So silanase is used to improve the nutritional and gut health, and then break down non-starch polysaccharides. It's included, I think, in all poultry feed used. Protease is not so often used, but it's included in order sort of to increase the protein digestibility. And this here all started with this as a project we had together with Side Plant Breeding, DLG, and Novozymes, where Novozymes came back to us and said, there's something wrong. We can't. Sometimes our proteases works and sometimes they don't. So in the first here they have added to a barley protease 200 units. It's perfect. Everything is fine. But then down here they took another one and were adding the proteases to another cultivar, and then they saw no effect at all. There was something inhibiting the activity of that one. So together we did a huge screen, and this is a variation in a lot of winter wheat, and the activity, and you see there's sort of really some that has, and this is the residual activity we have here. Some has a high residual activity after being mixed with protein from winter wheat, and some has a low activity. If you take the barley and the silanase, you also see a really huge distribution on the effect of adding a silanase to different barleys. So we studied that, and we of course had an idea what is going on, but I will actually present the feeding experiment, the data of the feeding experiment we did, because we did pick from these and multiplied them and did it in several generations and so on. One that inhibit a lot and one that doesn't inhibit basically, well, not a lot, but just a little bit, and then some cultivars in between. So we took 4 cultivars of barley and multiplied them, and then sort of from our predictions then, cultivar A has a high inhibition of the silanase enzyme, whereas cultivar D has a much less inhibition, and then there's something in between. So it started with at 7 days. They were using this in the feed, and already in the first after the first week there was a huge difference. So the bad cultivar they grew from 212.8 grams to 514.6, whereas in the sort of the one that we sort of supposed was the good one, they gained weight from 210.9 to 553.4, and the farmers are very happy with that early weight gain there, because they then establish sort of a healthy population of broilers. Yeah, and sort of the trend continued all over as in through the entire study. And according to the farmer, this was possible by choosing cultivar D instead of A. It was possible to produce his broilers he could cut down. I think he said he could cut down one day in the production time, which is of course important for him, but also in the feed use is actually important. So it's simply just using and selecting a cultivar which supports the activity of the silanase. We are currently working on finding exactly what enzymes we are dealing with. This is a protease story where we have where Michael on poster 14 presents how he knocks out specific inhibitors. And you see here is out here you have a non-inhibited enzyme activity, and then you mix in a wild type. You see the activity is decreased significantly. Here he is knocked out with his CRISPR different protease inhibitors, and there's a significant effect of it. We have done one experiment in pigs with chicken, with barley, and selected protease. To barley with 2 different levels of protease inhibition, and of course it's only 8 pigs, so the material is not so big, but there is an effect, a significant effect of using barley in combination, protease in combination with the right protease. So there's something in pigs there also. So the silanase knocks out. We know that we can knock out the specific silanase and then gain activity of the silanase added to the feed. So in conclusion, we know that now from what we have done that phosphate and calcium digestibility can be increased by improving the phytase activity in the feed, and by choosing the right combinations, a non-inhibited combination of cultivar and feed enzyme, you can actually get a significant increased performance of the broilers, both for silanases and in proteases for pigs. So in conclusion, feed barley and also wheat has the potential to improve the animal performance and reduce the secretion of non-digested nutrients into the environment. So there is basically a potential there to use. So I have few slides more because I think this was only on feed, but just want to sort of pay your attention to barley is super useful and interesting crop to work with. I worked with it for many years, and this is a focus from a project we finished together with in Iceland there's a company called Off Genetics that's using barley to produce pharmaceutical proteins and different proteins there. They have these huge greenhouses where they produce them in the grain. So we were having had a collaboration with them where we were basically trying to accelerate the expression of these particular genes that they were using, then they could get a higher protein yield. And actually, interesting, so we did it this is Mikael's PhD work. First, we took a barley mutant where the bee hoarding has been knocked out, and then basically crossed them with their production lines and saw that you can we can replace the hoarding with something else, in this case, a pharmaceutical protein. We have also been playing with the unfolded protein response. We have a pre-study where we saw some stress factors, and that's by mingling with them, you can actually accelerate the expression of a specific protein in the grain. It was useful here, but I guess also it can be useful in other cases where you want to promote a specific protein in the barley gene. So I decided yesterday to bring this slide also, because one day we had some materials where we had taken the phytase from cereals and expressed it in a barley. Then we checked after 3 years, we tested the materials, and there was super high activity already, and we actually learned that these enzymes here, the barley enzymes themselves, are super interesting. They can stay in it, and actually we're able to have up to 50,000 units in the straw and in the leaf of a mature material that has been stored for 3 years. And that's actually really, I know it's a GM, but I calculated you can use 20 grams of mature leaf material if you add per kilo per feet, and then you actually add 1,000 units, and that's what you need to get up in that. So it's sort of it's a super enzyme, but it's also a system where you could basically valorize the straw and the leaves substantially if you want. So last slide, then this is where we are. Research Center Flagerberg, and these are the people who has been over the years involved in this phytase internally work, and then I will stop here and see if there are any questions. Thank you.
The question is just, have you looked at any of the rye introductions into wheat on whether they, for the rye introduced chromosomes with the phytase genes, and whether they actually increase production in wheat? And there's also introgression lines as well with those chromosomes.
Yes, we have at some stage we did, and we haven't unfortunately been able to find one with a rye activity level.
Did you ever do RNA-seq on those lines to see if they actually were expressed in that context?
No, we didn't.
I found your comment on enzymes present in straw very interesting. So that was related to phytase, I understood. But did you check that in general? So I expected that, let's say, all the enzymes are either translocated or they are not active anymore. So,
but that's what surprised me a lot. They are fully active. So you take the straw and then you mill it, and then you put the buffer on, and then you can wash it. And then you have a full active enzyme there after 3 years storage in the straw.
But so the question is if that is only related to phytase, or if that's a general situation that there are plenty of still active enzymes present in. So for instance, also in regard to starch diet.
I can only say it's the case for phytase because we didn't do it with others, but it's a substantial leftover activity. So it's apparently very stable towards degradation.
Okay, hello everybody. Thank you for the possibility and opportunity to show some results of a project about assessment of winter barley for feeding quality. So we stay with feed quality, and I first want to explain why we try to breed especially for feed quality, because I think most breeding is just in terms of quality, malt quality or yield, but I hope I can show you that it might be interesting to set feed quality as a special breeding aim. of course, I will show you how the experimental setup of our project is, and finally I sum all up, and maybe there are some questions to be discussed. Okay, I think we have heard today a lot why barley and cereals are so important for feeding, and the most part of the produced cereals is of course used for feeding, and probably even if in Europe or especially in Germany the consume of meat is a little bit reduced, I think in the worldwide scale the consumption of meat is still increasing, so there's still lots of meat need for feeding barley. But the main reason we did this project was that since a number of years the European Union requires a special N management, nitrogen management, to protect groundwater against the pollution called by nitrate from agricultural sources, which resulted in the German Düngeverordnung, which means a sort of fertilization regulation for the N usage. And so we have breeding, we have, I think, two main ways to deal with this. So the one is trying to optimize the N efficiency of the plant with increasing of yield and protein in the grain, and the second part is to optimize the protein quality in order to increase the N utilization by animals, because the better the quality and the amino acid composition of the proteins, the more nitrogen you feed will be taken by the animal, and the less N comes out at the backside of the pig. So actually, our colleagues from the Institute for Animal Feeding came to us and asked if we see a chance to optimize the protein quality in feeding barley for pig nutrition, and this resulted in a research project which consists of 4 work packages I will show you now. So at first we tried to find out if there are any differences in amino acid composition in different barley varieties. So we screened samples from the official variety trials of Bavaria and analyzed for grain protein content, amino acid composition, and other nutrients in the grain. The second work package is quite interesting, but I think it would take too much time to include this in the talk, but I just mentioned because the data resulting from this part of the project are included in the genome-wide association study at the end. So we also grew a set of 80 genotypes in our phenotyping facility under 3 different nitrogen treatments, and analyzed the whole material for N content, grain protein content, and amino acid composition. But this is not the main part of my talk today. The main part of my talk is the work package number 3, where we performed field trials with 80 genotypes in total in 2 locations in 2 years with 2 different N treatments. And we analyzed this collection, of course, for grain yield, grain protein content, amino acid compositions, and all other traits you can measure on a field. And in work package 4, we tried to associate these traits to genetic markers using a high throughput marker chip. Okay, first we were interested to find out, are there differences in protein quality in the current available varieties we are just testing in our variety trials. And of course, it's not very surprising that there is a correlation between the cool protein content and the leucine concentration. The lower the protein content, the higher is the leucine concentration, and you can observe this in all types of varieties we tested. You can see the orange triangles represent the 6-row barley varieties. The blue squares represent the 2-row barley varieties, and with these green dots here, this represent 2-row barley varieties with a lower end treatment, which was actually used to produce winter malting barley with low protein content. So, well, this is really not a surprise, but we have seen that in a collection of quite closely related varieties which are currently grown on the fields of the farmers, we find differences in the protein content and in the leucine concentration. And so we were thinking that when we enlarge the variety panel up to 80 genetically diverse genotypes, we would find more diverse and more variants in this trait, which could be used later on for plant breeding. So we did these field trials in 2 locations with a total of 80 genotypes, 50 in each location, and 20 checked genotypes with 2 different end treatments. So the always signed as N-opt is the optimal end treatment according to the official recommendation for N fertilization, and 70% means that we have reduced the amount of N to 70% to have a lower N level, of course. Yeah, the yield data from these field trials on this panel of 80 varieties, and what was quite interesting to observe is that the yield doesn't fall down too much if you reduce the N fertilization by 30%. So the dark green dots represent the higher N supply, and the bright green the lower N supply, and there's actually no significant difference between those. So this is quite an interesting aspect for the agricultural practice. And in opposite, of course, the correlation is clear, the leucine concentration, which means the protein quality, is increasing when you reduce the N supply. So again, here the green with the higher N supply and the bright green with the lower N supply. And so this one outcome of these trials that we can reduce the N level and do not lose too much yield, but we increase the quality of the protein. And the other is that we really can see differences between the varieties, which gives us hope that we can really breed and select for improved protein quality. So we just, to show this to farmers and get an impression of different varieties, we put all these data in such a web diagram where you can see some interesting varieties from these trials compared by all the parameters we measured: yield, crude protein, phosphorus content, leucine, methionine, threonine, tryptophan concentration, and leucine total yield from the hectare. So you see there are quite nice differences between different varieties, and this might be the basis for breeding for this trait. Of course, we asked our colleagues from the animal nutrition the question, these differences we observe in leucine and protein content, are they relevant for feeding? So is the effort breeding for quality worth all the time and money we put in? And they did a calculation for us, and so this is just an Excel sheet which is provided by the LFL online, where the farmer can put in just his all his input, acquisition of animals, foodstuff, and other sources of nitrogen and phosphorus, and then it's calculated by some standard values for N utilization, and the farmer gets an output, and he can calculate the balance of nitrogen usage in his agricultural, in his farm. And for this purpose, we have here this calculation with two different varieties. Here on this side, you see the variety Frick with reduced N supply, and this variety has 88 gram crude protein in the ration, and this results over all different feeding processes in a medium for protein content of 129 grams. And using a different variety with more crude protein content and higher leucine concentration, you see you have a higher crude protein content. So what means higher? It means a difference of 8 gram in the average ration. This sounds quite very low, but actually, now I try to explain this to you. It's a little bit complicated, maybe. No. Okay, let's see. Now this is there's a classification of different feed ratios which is necessary to classify a farm for the emission of ammoniac from the stable. And you see you have here different feeding rations with reduced, with sinking protein content in the ration. And so you can see between this strong NP reduced ration and the very strong reduced ration, you have a difference also from 9 gram per kilogram. This is almost this what we have, what we could show between this varieties on the slide before. So actually, in the opinion of our colleagues from the animal feeding, if you use the appropriate variety and a quite intelligent N management, you can come down from one classification to another, which helps the farmers to stick to the legal regulations for ammoniac emission, and therefore helps them to keep their support, the money support from the government. So this seems to us a quite impressive result and motivation for breeding further on for the feed quality. Finally, we collected a lot of data from all our trials, and then it's quite a small step to perform a genome-wide association study. We did this with all different N treatments and all the traits we analyzed. So I just show the example for the leucine concentration, and you can see that it's distributed over the genome, some significant markers. So with respect to our small genotype collection, we just selected the one which lay above over the Bonferroni-Holm limits. And to show you one of those markers and its association to the feed quality, we have selected this one here on chromosome 5H, where you can see it's associated to the yield, and it's associated to reduced raw protein content, and of course, due to the correlation of all these parameters with an increased leucine content. Yeah, this is the current status of this project. Of course, there are some things missing. We have to evaluate a lot of the results, but we have already some evidence for the difference in protein quality of varieties, which could help farmers to optimize their N balance in agriculture. Of course, we have shown that the reduction of N supply improves quality, but doesn't reduce the yield too much, so that it's worth saving, especially at this time, a very expensive fertilizer. So in our opinion, it makes sense to care for protein quality in barley breeding and select for it. And we also have the tools for it because we have a near-infrared system to have a quite high throughput analysis and relatively cheap for the amino acids and other nutrients. And if this comes true, what we have seen in the genome analysis, we could develop markers for the selection. Of course, and this is still missing, we would try in the next round of this project to increase our genotype collection and evaluate the markers. And our favorite experiment would be a digestion experiment to see if really what comes out if we put a different variety inside the peak. Okay, so this was a short overview of our project, and I thank all the people who worked together with us. The project was funded by the Bavarian Ministry for Agriculture, and thank you for your attention.
I'd like to ask then, if you were to put forward a wish list to breeders and to plant scientists, to crop scientists, barley scientists, what would be on that wish list for barley, for feed barley, and developing feed barley?
I think it's very important to improve the amino acid composition because you have seen this topic is also very interesting for farmers. We have shown this the pig feeders and the pig growers and so on, and they are very interested in this because already they look for a variety where they can optimize the end circulation in the agriculture and reduce the total N amount they use. So I think this is for feeding barley quite important. But together we have many other very important problems like ramularia, which is I think the most important problem at the moment in barley. But on the quality and the usage side, I think this protein story is quite important.
Two o'clock, time for beer talk. Maybe a little bit early, but let's go for it. Yeah, I'm Christoph Doctor. I would like to thank the organizers for giving me the chance to present here for the Carlsberg Research Laboratory. And I would like to talk about trait development in barley. Before I do that, I would like to say something about Carlsberg itself. Carlsberg is a very large brewing company. We brew about 11 billion liters of beer per year. We reach 150 markets. We have 140 brands in our family, and we are 40,000 passionate people. Carlsberg was founded some 170 years ago by a Danish brewer, J.C. Jacobsen, and J.C. Jacobsen was extremely interested in brewing beer of the highest quality, and he was interested in the science of beer brewing to make the highest quality. So he actually founded the Carlsberg Research Laboratory some 150 years ago to investigate all processes of beer brewing. And we are very proud of our scientific achievements over the years, like Professor Emil Christian Hansen, who purified the first lager yeast, Kjeldal with his nitrogen quantification, Sørensen with his pH concept. And more recently, we investigated different ways of storing beer, like the Draftmaster brewing keg. And now, under the lead of Birgitte Skærhauge, we are looking very much into new barley traits, and we developed a new Find-It platform, which I will talk about later. So today, we are basically 4 main research areas. We have raw materials, which is primarily barley research. We have new ingredients, which is hops and botanicals. We investigate yeast and fermentation and the brewing science and technology. If you want to do trait development, we think that genomes are very important for sure. So we were always interested in genome projects, and we, from very early on, like in the 1990s, wanted to be part of genome sequencing consortia. So we were part of the yeast sequencing in 1992. We are a really happy partner of the IBSC in 2017, where the first Morex assembly was published. And now we are actually also looking in the hops genome, so we're sequencing hops. Our capabilities at Carlsberg for these brewing organisms are, like, we have the scientific expertise, we have the researchers to look into target identification for traits in barley, in yeast, and in hops. We have the breeding facilities, and we have the contractors that help us to grow crops, and we have the technology, and maybe most important, we also have all kind of brewing facilities to test these new traits and to test new beer brews, and we have a taste panel that gives us direct feedback on how these new beers taste actually. Generally, we are really happy about collaboration. So within Denmark, we have frame agreements on collaboration with the large universities like in Aarhus and Copenhagen, but then also in Europe and internationally. But let's talk about new traits and raw materials, and specifically barley. So Carlsberg was over the years very interested in quality traits for the beer brewing. But now we are becoming really interested in climate tolerance. I mean, we had really interesting talks yesterday about climate change, and especially what Brian said, this picture with the lonely beer drinker, this actually really concerns us. This is nothing we want to see in the future. So we want to investigate climate tolerance. We are interested in specialty barley, but also as blueprints, blue sky projects, we are looking into sorghum and wheatgrass, for example, as crops for the future. Over everything that we are doing now is the buzzword sustainability. The brewery is very concerned about our carbon emissions. So here you see the carbon emissions across the brewing value chain. And if you calculate, it's about 40% coming from agriculture, from malting, and from the brewing itself. And this is actually where we, as barley researchers, can come up with new traits to improve the carbon footprint of the brewery. If we look specifically into this process, talking about agro traits like yield robustness, climate tolerance, regenerative farming becomes more and more popular. Within the malting, germination efficiency is really important. Like pre-harvest sprouting, for example, is horrible. We don't like that at all. Then the enzyme optimization of the brewing enzymes, starch composition, filterability of the wort, different types of off flavors you can get during the beer brewing process. But then also, in the end, the whole drinking experience, like the freshness of the beer, the foam, the taste, and its shelf life, like that it doesn't develop any off flavors over time. Some of the more recent innovations that we now roll out to third parties, like third-party breweries and malteries, is our Nulox barley. So we have the Nulox 3G barley and the Nulox 4G barley, which have loss of function variants of the LOX genes and the MMT genes. The lipoxygenases are important to have to get rid of the T2N, this cardboard off flavor, which develops over time in the so it's basically an elongation of the shelf life. The MMT gene, that's really important to have null DMS. DMS is another off flavor which you get during the wort boiling, and you have to basically boil the wort very long to get rid of it. And if you don't have this gene, then you can basically save energy by not boiling it so long. So these are recent improvements that we made to our brewing barley, and well, it took quite a long time to develop these barleys, and we were looking for a way to accelerate this process. And as a brewery, we cannot use GMO, so our consumers wouldn't accept that. What we did, therefore, we invented Find It, which is an accelerated and we think a traditional trait breeding approach. For the sake of time, I will not go into the full details here, but you can read all about it in our publication now. It's basically a mutagenesis approach where we did like sodium acid mutagenesis with a rather low mutagenic treatment, because we didn't want to get too many mutations per individual. We then grew the M2 and M3 grains, and we actually sequenced the M3 grains, whole genome sequencing, to get the mutation profile. and to see what type of mutations we got. And as expected, we got mainly transitions here. And then we made pools of about 300 plants, and we started to collect the DNA from these pools. And this is a little bit the trick of this technology, that you first make the pools, then you make the DNA extractions. So it's this, this pool split approach that we did. So by doing so, you can actually build very large populations of up to 500,000 mutant plants. That's what we did on Planet. And by using digital PCR, you can basically find the pools with the specific transition of your interest. And this is going really fast within a couple of days. With this approach, you can find all kinds of knockout variants, amino acid exchanges. We also found promoter and mRNA variants that give you a higher transcript abundance. And it's a species-independent approach. It's very flexible. Like, you can use all kinds of species for it. It's really fast. And if you use this low mutagenetic treatment, then you can actually use the plant directly in the breeding pipeline. Just to comment on what Henrik said before, so the brewery understood how interesting this technology is, so they formed this new research platform, which we call Tratomic. And Tratomic is using the FindIt to explore all kinds of organisms that are not brewing related. So the Carlsberg Research Laboratory itself will do all the brewing related organisms like barley, while Tratomic will do the rest. So just said that we can find all kinds of knockouts. So for the publication, we actually isolated 100 knockout variants of our interest. And among them were the usual suspects that we all know about, like the newt barley, like row-type barley. We also targeted barleys with a low beta-glucan content or with a waxy starch. And one interesting one was the grain weight 2 that is known from rice. So there's no knockout barley mutant known so far. So we knocked it out and we got the wider grain as in rice. So it's a very nice, nice approach to work with. What we can do is, because it's non-GMO, we can take these barleys directly on the field to make like a pre-evaluation. So here you see a Paustian wild type, in this case from a Paustian library. And this here is a Della mutant which is a GA gain of function mutation. You can see it's a little bit earlier flowering. It has taller stems, it has a longer spike, and it gets a larger grain. So when I talk about accelerated trait breeding with a purpose, I come up with an example here for the grain beta-glucan content. So the CSLF6 gene, if you knock it out, we know that we have no beta-glucan in the grain. And what we didn't know from the literature is, when we take this plant and put it to the field, what is its agronomic performance. So we did that, and we could see that we got some 30% lower yields actually. And it's a very interesting trait because it improves your world filterability. But if you get 30% lower yield, you cannot use it in large scale to raise it as a cultivar. So what we did is we modeled the protein, and we looked into a whole lot of different type of amino acid exchanges. And then we identified 3 amino acid exchanges that have like 50% lower beta-glucan content, but which all get the wild type grain yield. And those we took and we started backcrossing them into elite barley. And from the grain identification of the single mutant to the backcrossing to the registration of a cultivar called now Selina, it took us 4 years to accelerate it to the 100-ton level. So just as an example on how we envision to do that for brewing traits in the future. So the other thing that we are very interested in from now on is the climate targets. So we just started a new program within the research laboratory on increasing the robustness of elite malting barley on yield. We are very interested in drought and salt tolerance targets, and in general to discover all kinds of climate adaptation traits from serial pangenomes and test them then with the Find It technology directly in elite barley. The goal is to keep the barley malt quality high, but to get a competitive yield. And as an example for a project that we started some 3 years ago, with Dominique Vico from Secobar Researches and also Pierre Penn, was on pre-harvest sprouting. So pre-harvest sprouting is a general problem for wheat and barley and causes some $1 billion in loss per year. And it's obviously very important for malt quality. So we were looking into pre-harvest sprouted plants, and we identified the key mutation for pre-harvest sprouting in North American malting barley. And we think we also identified we have a really strong candidate for pre-harvest sprouting in Ethiopian barley. And to prove our hypothesis now, we developed a couple of Find It mutants and test them in the field and for pre-harvest sprouting. So Find It beyond barley. So as I said earlier, it's a very flexible system. So you can use all kinds of other crops as well. And therefore, we started a project called Crops for the Future. So one of these projects is accelerated domestication of sorghum. That's a collaboration with the University of Queensland and Copenhagen University. And it's about perfecting sorghum for food and brewing. So we made our first Find It library in sorghum and now started screening for mutants here as well. Then we did another project on wheatgrass. Wheatgrass is a very interesting perennial with a very large root system, which is obviously interesting for drought stress and or drought tolerance. If you see here on the side, you see a wheatgrass plant beside a bread wheat. And the goal is over the next years to use the Find It to quickly domesticate it and hopefully get closer to the stature of the bread wheat to increase the yield. And yeah. And the last project that I want to talk about is about rice. So we have a collaboration with Eske Willerslev at the University of Copenhagen and then the University of Cambridge, who is sequencing the ancient rice genome. And we just developed our first rice mutant population for this project in Kitake rice. And that's it from my side. I hope I could give you an overview over what we are currently doing at the Cartwright Research Laboratory, and happy to take some questions.
Thanks a lot, very interesting. Have you had a chance to estimate already what proportion of genes you have not been able to identify mutations for? I mean, like given that you cannot recognize everything normally, with AMS there are some genes that are, say, recalcitrant to mutagenesis by chemicals. So have you estimated how many, or the frequency of genes you have not been able to knock out or to find mutants for?
Well, we whole genome sequenced several of the mutants, and this gave us basically the mutation spectrum that we got for transitions versus conversions, and basically how likely a certain transition is concerning the neighboring amino acid, nucleotides, and therefore we got a really good idea what type of mutations we can target. Is that what you're about to ask, or?
Let me phrase it differently. Have you found a gene that you've tried to target and you haven't found mutations for?
No, not for a knockout. We always have, we had it that we couldn't find a specific knockout in one of our libraries, but then we designed a new knockout, so to speak, a different tryptophan to stop, for example. Each barley gene has multiple tryptophan to stop possibilities, and if you don't find one, you can just go to the next one. Then it's in the end, it's only dependent on the library size that you choose to do. We could easily scale it up to make it even larger, and we actually decided now to increase it from 500,000 to 800,000 barleys just to have an even higher possibility to find a certain amino acid exchange or knockout. Yeah.
Thank you for your talk. Can you please elaborate a little bit on the screening of the digital PCR? Because when I'm right, you have only 1 or 2 bases where you can screen. So how can you screen many bases or many targets or many mutant points?
We just screen for a single mutant point. So it's a standard Tagman assay with 2 fluorescence markers. So we target one specific SNP, but since the libraries are so large and since we know the mutation spectrum, we can be rather certain that this SNP is within our library. We just have to find it, so to speak. So we start with the very large libraries, and then very large pools, and then we go to smaller pools. But it goes very quickly. It's a very fast screening. Well, we could target other mutants, yes. I'm not sure if we missed them, but it's just you decide what you want, right? Hi, Robbie.
Nice talk, nice platform. I just wonder if you could elaborate on a couple of the mutants that you mentioned in the talk. Both of them would probably affect malting quality, but you didn't talk about it at all. So you talked about the MKK3 mutation, and you talked about the the CSLF6 mutations. Particularly the latter one, where you've got it through into the field. What does it perform like in the malt house and in the brewery?
Well, we scaled it up to 100 tons because it's working. So we had really
All
good
barley will work. Is it better?
Yep, it had really good results in the filterability. So therefore, we decided to breed and register a commercial line in it.
So you'll start to use it?
Yeah.
So how much would be the lifetime of such new variety, based on your experience? If you get it into the workflow, how long is it going to last before a newcomer will come and replace it?
That's a good question for the commercial people in Carlsberg and also for the breeders here. But I mean, we constantly have to improve the yield of our varieties. If our Carlsberg varieties with specific brewing-related traits are outperformed by other commercially available varieties, then our supply chain procurement is not very interested in anymore. So we have to constantly keep up with the yield. So we have an elite yield breeding program for each of our Carlsberg quality lines.
Thanks, that was a nice talk. You increased the population size dramatically compared to classical mutational breeding in order to get rid of extra mutants at the same time. So what is your experience about mutational load? So how many genes of type you have giving rise to, let's say, lower performance in other traits you are not concerned? And second, how you design the chemical you use, is it always the same, N2, for instance, or EMS, or is it depending on the mutation you want to have? So amino acid substitution, you use different chemicals to produce them.
Start with the second question. So we use sodium acid. And we are very interested in testing different mutagens to get a different mutation spectrum, for example, to increase transversions, which would then lead to possibly other amino acid exchanges that we could target. But we haven't tested it yet. And concerning the mutation load per individual, so that's actually the nice thing about this platform. You can decide, depending on what the purpose is of your population. If you want to do elite breeding, you don't want to have too many background mutations. So then you need to have a large population. So when we did two planet populations, one was more, let's say, for functional gene analysis, and there we induced an average some 15,000 point mutations per individual, and the one for breeding is about 5,000. If you do the breeding calculations like with the low mutagenesis, you get very quickly rid of these background mutations just in elite breeding. And we did some number games with let's say, the amount of natural variations you induce just by normal crosses, and it's actually huge numbers compared to 5,000 background mutations. So we're not really worried about that. Also in the field, when we grow them in the field, I mean, we make yield plots. We make like several repetitions of yield plots, and we see directly if they perform or if they don't perform. If we have like very interesting trait which performs really bad in the field, we directly start backcrossing it, or we just isolate a new mutant to test it again.
Yeah, thanks. I liked your find it pipeline. What I did not quite understand is how do you know what mutation you actually need to target? In particular, when it's not malting quality, but climate resilience. Or do you just use that then for using forward genetics rather than reverse genetics?
So I mean, the whole genome sequencing for the sodium acid population told us the mutation spectrum. So therefore, we know how to design the probes. Is that your question?
No,
Okay.
how do you know which mutation is actually relevant to
Yeah.
improve your lines?
And that's the
Especially for
difficult one,
climate
yes.
resilience.
Yes.
How do you do that? That's the big secret. You're not telling us, so.
No. If I would have the super answer for that, I would tell you. But no, I mean, we are very interested in investigating that, and especially the climate resilience. That's a very complex topic, and that's actually one of the reasons why I'm standing here presenting you to find it, because we would be very interested in your input from the community to this topic. So we should discuss that.
So currently it's taking me particularly to have a homozygous mutant using CRISPR about 8 months. So with your pipeline, how long does it take you from, let's say, from the beginning of mutagenizing to having your mutant in a homozygous state?
Well, the mutagenesis and growing the crop over 2 generations, so we would typically do the mutagenesis in fall, send the mutagenized strain to New Zealand. They would harvest the first population for us, would send it back, and then we make the pools in July the next year in Denmark. So the design of the population library takes 9-10 months, but once you have it, then you can screen it for a long time. So, and if you target the right, let's say the right nucleotides that you know from your whole genome sequencing, then this whole process is extremely fast. If you have developed a very new library, it takes you a while because there's one bottleneck step in the pooling. You have to go back to the grain pool and you have to basically drill holes into the endosperm of a grain, which is non-disruptive, so the grain will not die from it, and you can actually store it in the freezer for several years. This is how you design your subpools, or how you develop your subpools. And we are lucky because we have a breeding program. We have a large amount of student helpers, and they can drill about 1,000 grains per work shift per person. And once you establish these subpools, you can store them in the freezer as well. You can use them for years, and then it's really quickly. Then you design the probes, you wait for them to arrive, and then it's a couple of days screening process until you have your grain and you grow it and validate it. Yeah.
Mr. Chairman, dear colleagues, good afternoon. First of all, I would like to thank organizing committee to allow me to present here the results of our several years work. Several years ago, we had a project where the task of our team was to develop molecular markers for malting barley breeding for Czech beer. Czech beer is a protected geographical indication that this indication was get in 2008. It is characterized by specific raw material, malt and hop, and specific technological process, which are characterized by decoction mashing, bottom fermentation, and long ripening time during months. What is characterized Czech beer is with final extract and lower achievable degree of fermentation, more intense color of the wort, higher bitterness of final product, higher pH, and higher polyphenol content. The first question we had to answer was if there are any differences between barley genotypes on technological level and how it is at the genetic level. For that, we selected 8 barley varieties, 4 of them suitable for Czech beer. We named there or marked them with C, and another 4 malting barley varieties suitable for another type of beers. We marked them with E. We added also 50 breeding lice into the analysis. These genotypes were malted and malting quality parameters were determined. ANOVA analysis showed that which parameters are significantly different between these two groups of barley, C and E. They are malting quality indicator, relative extract at 45 degrees centigrade, callback index, achievable degree of fermentation, beta-glucan content in wort, and color of wort. We did discrimination analysis where technological parameters were taken into the model, and we got the discrimination power 0.2021. Then we did genotyping using microsatellite analysis at the time. We use 43 microsatellite OC and got 236 alleles. Cluster analysis was done in Darwin software, and it showed not really straightforward discrimination between C and E barley genotypes. Then we did the exact test of population differentiation, and we found that there are differences. These two groups are really different on the level of probability 99%. These results were supported by analysis of population structure. And you can see in this table that C and E varieties occupied or are involved in different K clusters. We repeated discrimination analysis where not only technological but also genotyping data were involved, and we got discrimination power 0.06. You can see on the picture that varieties of the group C and of the group E were discriminated, were separated each other. Then we started a huge sequencing. At that time, we used Sanger sequencing and started with some genes of amyloidotic complex. But unfortunately, we found no sequencing differences between these two groups. We found surely differences between varieties, but not between these two groups. On the other hand, we found differences in specific microsatellite locus. It is BMAC 120, and this sequences was blasted, and we found that it is some transcription factor. There are two possibilities here. We found differences like SNPs and also the own microsatellites. These differences were used for developing our first marker. We found three. You can see now two of them. One is based of SNP, and the second one, codominant, is based on a microsatellite repetition. These two markers or three markers were used in barley breeding. In the last year of the project, we would like to speed up our search for differences between these two groups, and therefore we selected transcriptome sequencing using NGS. For that, we did malting experiment with previously selected barley varieties, and we collected samples during the malting process. We extracted RNA, and because of money shortage, we don't have enough money. Therefore, we selected only 3 varieties per each group and only 3 data point, T0, so the third day of soaking and the third day of germination. For NGS analysis, we selected Illumina platform, and we had sequenced our samples by a SeqMe company. As we got our sequences several months before Morex reference was released, we had to prepare our own reference. For that, we selected Trinity software. We used Trinity software, and besides of our sequences, we added also sequences of Hordeum species, Oryza sativa, Brachypodium species, and Swiss prot database. Then we do mapping against our own reference. And we used our sequences for 2 analyses. Firstly, we did variant calling, and after this pipeline and filtering and selecting some special contig with suitable, especially with suitable quality and other our purposes, we got finally 22 contigs with 33 SNPs and 2 indels. These sequences were used for proposing new molecular markers. Some of them undergone optimization and validation and can be used in barley breeding for Czech beer. The second analysis, the most important, was the differential expression analysis. We found in total 227 differential expressed contigs. And then we did blast analysis of these contigs. In cluster analysis, you can see that these differentially expressed contigs cause the differences between these 2 groups of varieties, C and E. After blasting, we found several genes, and these sequences were used for primer design, and then we did transcription profiling using real-time PCR. You can see some of our results. In future, we would like to firstly remap our sequencing against Morex references, because we would like to map our genes and probably find some others. And the second one, we would like to continue with transcription profiling, because we had much more samples collected during the malting process. That's why we would like to continue with this work. At the end, I would like to thank my colleagues who were involved in our work, and thank you for your attention.
Did you validate these 3 SNPs or 3 markers in Czech type and non-Czech type malting barley, and they were all the time correlated, or what?
Yes, we use all malting barley that were at the disposition. We collaborated with Czech breeders who have their own collection, and we used this collection for the validation.